Picking up from the last video, to come back to this idea of an ACE inhibitor. Now, ACE stands for angiotensin converting enzyme. So the inhibitor is blocking angiotensin converting enzyme, which means that you reduce the amount of angiotensin that's produced. Now, angiotensin is a hormone which is very, very important in the kidneys in particular, but also various other parts of the body like the lung. And angiotensin is a hormone which acts on the smooth muscle of blood vessels to cause constriction. And if you cause constriction, then the diameter here of the blood vessel is narrower, and the angiotensin is going to cause an increase in blood pressure. So an increase in blood pressure is essential for vital kidney function, but you don't want the blood pressure being too high. So if you've already got elevated blood pressure, then you might be prescribed this ACE inhibitor, which will prevent this enzyme from converting the enzyme as part of what's called the RAA system. You don't need to know the details of that, but you need to appreciate that angiotensin is going to cause vasoconstriction. Angiotensin is produced as a consequence of angiotensin converting enzyme being able to function. If you block that functioning, then you prevent the angiotensin being released, and you prevent the constriction. So the blood vessel remains dilated and blood pressure is consequently lower. So this is used for the treatment of people with high blood pressure. Just repeating what we've already met is this effect on enzyme kinetics, and making sure that you appreciate the features of a competitive versus a non-competitive inhibitor are different. And here you can see what's referred to as a half Vmax. This is slightly new. So Vmax is the total maximum reaction that can happen if the enzyme is inhibited. And what you could do is you could look at different inhibitors being present, and you can look at this value half Vmax. You can also look at the same enzyme. To just make it that clear, half Vmax means it's half the maximum velocity, and then it's the substrate concentration, which is half that maximum velocity. This is sometimes called Michaelis-Menten constant. But what you can appreciate is if you look at academic literature, they may ask you to think about comparisons of half Vmax. So half Vmax is giving you an idea of either the same enzyme in different species and its effectiveness, or the presence of an inhibitor and how that might impact it. So that's just an addition. So these slides are found in the PowerPoint, which is within the Google Classroom. This again is end product inhibition, which we talked about in video 2. This video, if you've not already watched it, is very well worth doing. Make sure that you've got the idea of the end product blocking the very first enzyme at the allosteric site. Here again is a summary of the example that we met in terms of the amino acids, and to finish off with this idea of bioinformatics. And here it's very well worth again looking at BioNinja. Now the example that the syllabus asks you to look at is looking at anti-malarial drugs. So malaria is the disease, and the malaria disease is caused by a protozoa species called Plasmodium. Now the Plasmodium can host itself in the female mosquito's salivary gland. When the female mosquito then goes on to bite a human, it injects the Plasmodium into the human, and that then impacts the red blood cells of that particular person. Now there are different species of Plasmodium, and some of them are very, very dangerous, and others you can recover from, but these protozoa can stay in your system. So it's a very nasty disease. I'm not sure, it used to be the biggest killer of humans in terms of disease. I'm not sure if it still holds that title. But what we need to understand here is that the parasite, the Plasmodium, needs to infect a human cell and a host cell for it to remain intact. Now, what bioinformatics aims to do is it looks at various different types of experts. So it's going to look at, for example, biologists, which are going to take the genome and they're going to code for the genome. They're going to look for the proteome. And if they know the proteins, then they can identify the enzymes. And by identifying the enzymes, they can maybe start to think about the type of inhibition that can be used to combat that enzyme, which then stops it from the plasmodium, stops the plasmodium enzymes from functioning properly. So there's a range of approaches which are in place, but this idea comes up quite a lot in your course because it's really the nature of science, right? How we use databases, so databases of the genome sequence of the plasmodium, databases of the proteome sequences, and how we can then look at either synthetically creating or looking for an already identifiable compound to block that particular protein. So again, these two slides are there for you to take a look at. Plasmodium falciparum is the most deadly of all. And here it gives you some links and connections to look at various different lab work that can be used to measure enzyme inhibition. So this is worth looking at, and that's it for 8.1. Can I ask you to look at all three of these videos? Can you work through the booklet, work through Bionology, make sure that you've got notes, and come back, obviously, and field as many questions as you need to get a secure understanding.